coming. I'm Yoder from the University of British Columbia, and I'm going to be presenting to you today some really pretty preliminary results on the ecological genomics of parallel adaptation in some tree species. There we go. So the key question of this talk really is, does evolution repeat itself? If we started from scratch with an empty planet and life beginning all over again, would we eventually end up with a world that looks very much like ours, with intelligent life and possibly bipedal speaking apes, or would we come up with something very different? That's sort of the extreme version of this question. In pop culture, this often turns up in time travel movies. So there's sort of the 12 Monkeys model, where you send somebody back in time, like poor Bruce Willis here, to try to prevent a catastrophe. But history is predictable, and there are forces beyond his control, and so everything happens all over again, and he has no effect on it. And then there's also the model that you see in the Back to the Future movies, where history is much more contingent and reliant on small forces. So I haven't seen that set of movies in a while, but I think I remember correctly, somebody's parents didn't get together, and then Donald Trump became president. It's bad. In real biological systems, of course, we know that there's lots of examples that sort of span this range. These are just cartoonish extremes. But what we are interested in is how predictable is adaptation. If we see adaptive change in a particular gene in response to a particular set of selective conditions, can we know when we go to another population of the same species or another species that gene is also likely to be a target of selection under the same conditions? And knowing that is the case, that sort of assumption and our experience that it does happen underlies a lot of work in medical genetics and conservation genetics and genomics, and translating functional information between species. So the Adaptation project, which I'm a part of, is really collecting data that can help to inform this big broad question. Adaptation is looking at parallel adaptation to climate in 3 tree species across British Columbia and Alberta, which capture very different kinds of potential parallelism. The 2 species studied are Pinus contorta, lodgepole pine, the map on the left of the screen here, and Ecological and Engelmannii, white and Engelmann spruce. They have, as you can see, similar ranges across the 2 provinces. They occupy broadly similar ranges of climates as well. And pine and spruce have been on separate evolutionary trajectories. They last shared a common evolutionary ancestor about 140 million years ago, maybe more. So they have had lots of time to do different things, and if the same genes are involved in adaptation, that indicates deep parallelism. The spruce species separated about 5 million years ago, and they've had long periods of hybridization over that time, and they currently have lots of hybrids along climactic gradients. So they are adapting to climate from, to some degree, a shared gene pool, or a shared pool of genetic variation. The project has been looking for genotypes associated with 22 biologically informed climate variables estimated across the native ranges where the samples were collected, and 17 common garden phenotypes. And if you were in the morning session, you saw Sam Yeaman talk about the results from sequence capture-based work with hundreds of individuals from the sample sites that I showed you on those previous slides. This was targeted diagnosis of single nucleotide polymorphisms, targeting exons, so gene-focused, but also intergenic regions for neutral controls. And that gave us a first look at the genetic basis of adaptation to climate variation and the genetic basis of climate-associated phenotypes. Now I'm working on the next stage of this project, which is using SNP genotyping arrays that are collecting genotypes at tens of thousands of single nucleotide polymorphism markers, genotyped in thousands of trees across those same ranges. The genotyping array was designed based on the sequence capture results, so that we have SNPs on the arrays that we know showed associations to climate and the climate-associated phenotypes in the first analysis. But we also have neutral SNPs to give us a genome-wide control background. And because we have a larger sample, we can start to make comparisons of parallelism at different scales with this data, and that's what I'm going to try to show you today. So just as a general orientation, a very cartoonish view of what I'm doing with this data is looking for genotype environment associations, going from doing an analysis that essentially goes from SNP to SNP and asks whether individuals who carry one allele at that SNP tend to be in different climates from individuals who carry a different allele at that SNP. And the specific method I'm using for this is originally designed as BayNB and as implemented in a program called BayPass, which provides model-based controls for the effects of population structure. So we know that there may be confounding due to relatedness across the landscape when we're looking for these associations with spatially distributed things like climate. And so for each SNP in the data set, BayPass returns an estimate of strength of association called a term called beta, which you'll see mentioned in some of these outcomes. And what I can do is compare the strength of beta association at SNPs and across multiple SNPs on a gene in different groups of samples, either between pine and spruce or between the two spruce species, to ask whether we see similar signals in those two groups. So here's what we get for that pine versus spruce. Again, this is working with samples at all of these sites across the ranges of the two. And so for these, this is restricted also to genes where we have one-to-one orthologs identified in the two species. So we have pretty good ideas that we're looking at the same chunk of sequence across that 140 million-year gap. And this is the association that regression of those beta terms, the strength of climate association, for all of the SNPs in the data set in spruce and in pine for a variable called climate moisture deficit, which is a measure of potential drought stress in the environment. The lighter points are the genomic background SNPs that we don't have any a priori expectation are associated with climate moisture deficit. The darker points are the SNPs that showed association with CMD in the previous seq capture analysis. So those are our candidates. And again, this is all pretty preliminary stuff. But the association that you see there is statistically significant and actually strongly statistically significant, but it's pretty weak and it doesn't explain a lot of variation. In the background, there is no association. In the candidates, there's this significant positive association. And if we look across all of, so we see weak parallelism. If we look across all of the different climate variables that we've been looking at in the study, we see that differentiation between the background, the lighter points, and the candidates, the darker points. Now, this is the correlation term and the 95% confidence interval on that correlation term for these different sets of SNPs at each climate variable. And so you see the background SNPs are generally staying around 0. We don't see significant associations. The candidates are generally overlapping 0 with their 95% confidence interval, or they're positive. So here's climate moisture deficit. Oh, and climate moisture deficit's annual heat moisture index, which is correlated with CMD to some degree. That's why they're close to each other on that axis. Here's the extreme maximum temperature. That also shows some parallelism, and extreme minimum temperature over a 30-year period. So it looks like for this very deep comparison, we're in 12 monkeys world, where the things that we're able to make the comparison across that 140 million year gap are underlying similar responses in spruce and in pine. What about when we look at the much shallower differentiation, spruce versus spruce? So this is something we were able to do because we have this much larger data set with the SNP array. Again, in a lot of cases, we see positive associations in the signals at individual SNPs in the two spruce species. So this is Glauca versus Engelmannii, and I've excluded hybrid populations in this comparison. And so this is strength of association to the mean temperature in the coldest month. Again, candidates for that from the CCAP show a positive association. The background doesn't. But if I go and do this again for all of those different climate variables, we have positive associations in the candidates like MCMT that I just showed you, but there's also some interesting cases where there are negative associations. So this is the summer heat moisture index, which is another drought stress related composite variable. And this is, if you look at scatter plot, this is again not explaining more or less variation than we saw in the positive associations, and it's a statistically significant negative correlation. If I break down the beta terms plot the beta terms in each species against each other, for these different variables where I do see this negative correlation, I can see cases like summer heat moisture index or like mean annual temperature, where things that are the SNPs that have the biggest differences between the two species seem to have sort of equal and roughly opposite on average effects in the two species. The beta terms are crossing. But I also see things like the number of cross-breed days here. SNPs with big differences go from having a big effect, positive or negative, in Engelmannii to having on average, roughly neutral effect in Glauca. So there's cases where we may be seeing differences in strength effect, but also cases where we may be seeing differences in quality of effect. So this looks, again, a little more like the opposite model, like everything is random and contingent on who's doing the adapting. Possible explanations. Well, this could be statistical noise. This is still early enough in the process. I may continue to work with possible explanations and come up with something that gives me a completely different graph 2 weeks from now. It could be that we're seeing SNPs in the two species that are linked with different functional variants. It could also be that I'm looking at actual functional variants that have different effects. And fortunately, I've got a lot of tools yet in the kit, a lot of available data to continue to corroborate what I'm seeing in this initial pass. So we have differential gene expression data for many of the genes that are being genotyped on the SNP array. We have the common garden phenotype associations, which are just finished running before I had to submit this talk. So that's the next thing I'm going to look at when I get home. And I'm working on building linkage maps for pine and spruce. We have controlled crosses that will let me start to do that, so I can maybe get to that question of whether we have linkage to different variants in the two species. So does evolution repeat itself? Maybe over the long term it does. It's where we're able to make those comparisons. We see deeper forces acting on what's available to be adapted. Over the short term, things are a little more random and contingent. But stay tuned, because there's a lot that I have to do yet with this. So with that, I want to thank the Adaptation collaborators, particularly Sally Aiken, my postdoc advisor, and Sam Yeaman, who's been a big help getting me set up on this work. And I believe the second bell has rung, so I've got some time to take questions.